Hello everyone and welcome to Instant Biology by Dr. Nilab. And today what we are going to do is we are going to discuss a very important topic of immunology lecture series. It has been a while since I have made a video on immunology. So I would be today making a video on immunology, and I would be moving forward talking about B cell maturation and activation. Okay, what I'll be talking about is B cell maturation and activation. If you have seen my previous videos, then you would understand that B cell is the part of the humoral immunity, and in order for B cell to function properly, it has to undergo maturation. Okay, and how does the maturation take place? Where does the synthesis of the B cell take place? We would be talking about all of those things, and this is an important topic if you talk about the CSIR net or GATE biotechnology examinations. So let us, without much ado, let us talk about this topic. So B cell maturation and activation, this B cell maturation can be divided into multiple steps. For your easy understanding, I have divided it into multiple steps. Okay, so before talking about maturation, let us first of all understand that what is the site of synthesis. That means where actually does this B cell get synthesized. Okay, so synthesis as well as maturation in case of B cell, remember when we are talking about B cell, both synthesis and maturation would take place in bone marrow. Right, so synthesis and maturation takes place in bone marrow. So this is an important concept and has been asked in examinations, so you should at least remember this, that both synthesis and maturation takes place in bone marrow. But you would understand this, that bone marrow actually develops after a particular age, because when the bones get large and tough, so larger bones or the bones that are greater in size, just like femur, they inside them have this bone marrow. But you would be able to logically also understand this, that bone marrow cannot be present before birth, right? When the mother is in the womb, when the baby is in the womb, a mother's womb, then this bone marrow would not be present over there. That is why I am saying that at that particular instant, when the bone marrow is not present, when the baby is still not born, that means before birth, the function of this bone marrow is performed by yolk sac or fetal liver or fetal bone marrow. What do we mean by fetal liver or fetal bone marrow? That means that the liver of that individual, the small fetus that is present, or the bone marrow of the fetus that is present, that synthesizes actually the B cells. Okay, now let us talk about what is the process or what is the mechanism through which the development or maturation of B cells take place. Okay, so first of all, let us talk about that where does the differentiation or where does the process start from. I also have a diagrammatic representation over here, which I will go to after the text that I am mentioning is completed. Okay, so as to make my points more clear. So the first step is these lymphoid cells, not lymphoid but lymphoid stem cells I am talking about. These lymphoid stem cells, what do? They undergo differentiation. Okay, what does the differentiation means? Differentiation means they would gain a particular fate. Okay, and now they, after gaining the particular fate, after deciding that they would be performing this kind of a function, now they are called as progenitor B cells. Okay, progenitor means it is one stage previous to that of the precursor cells. That means precursor will come over here, and just before precursor we have another stage that is called as progenitor stage. and in short this stage is abbreviated as pro cells. Okay, in this is called as pro cells. And what happens inside the pro cells? In case of pro cells, heavy chain rearrangement takes place. Okay, now you would be knowing what are heavy chains. Heavy chains basically are the greater sized chains that are present. You would be knowing that there are light chains and there are heavy chains. So over here I am talking about heavy chains. If you do not know what are heavy chains or light chains, I would request you to go and check out the previous videos so that you have a clear idea about that. So heavy chain rearrangement takes place over here, right? And after that what happens is bone marrow stromal cells. Now these are some very important cells that have very particular role that they play in order for the conversion of or for the maturation of progenitor B cells into precursor B cells, right? So progenitor B cells are converting into precursor B cells because of the role of bone marrow stromal cells. Now let us understand what do the bone marrow stromal cells do. These bone marrow stromal cells, what do? They come in contact with this progenitor B cells. They are in contact with the progenitor B cells. That means they have adhesive interaction. Okay, they interact with each other, and because of this adhesive interaction, so one thing that they do is adhesive interaction, and the second thing that they do is cytokine production of cytokine interleukin 7. Okay, so because of these two things, what happens is the progenitor B cell matures into precursor B cells, and it is abbreviated as pre B cells. Progenitor was abbreviated as pro B cells. Now after reaching this precursor B cell stage, there are multiple events that are happening in this phase. Let us talk about this. Okay, now as I've said that in the progenitor B cell itself, the heavy chain rearrangement has taken place. Okay, but and because of the heavy chain rearrangement, we have IgM, mu heavy chain. Now you would be knowing when the mu heavy chain is present, it leads to the formation of IgM. Okay, so now this IgM is present, but the light chain is yet not decided. That means the rearrangement of light chain is yet to be completed. So in this particular stage, or in this after reaching the precursor B cell stage, mu heavy chain would be present along with the lambda 5 and VPRE-B proteins. Now what are these? Lambda 5 and VPRE-B proteins, these are surrogate light chains. What does surrogate light chain means? These are structurally same to the kappa and the lambda light chains. You would be knowing that when we are talking about the light chains, there are two types of light chains, kappa chain and the lambda chain. But before reaching to the kappa and the lambda chain, what we have is lambda 5 and VPRE-B proteins. These are surrogate light chains because they are structurally similar to the kappa and the lambda light chains. But one thing is very important, they are present, or these lambda 5 and VPRE-B proteins are present in all the B cells. They are invariant. There is no variance or there is no change in any B cell if you are talking with or with respect to this lambda 5 or VPRE-B proteins. Okay, because after this what will happen, the lambda the light chain rearrangement will take place after this particular stage, and after that the kappa and the lambda chains would be present. But for now, because in the initial stages we are just having VPRE-B and lambda 5 proteins. Okay, now after this what is happening is productive light chain gene rearrangement occurs. Now this is what I was talking about. Okay, now after reaching this stage, productive light chain gene rearrangement occurs. So just like the heavy chain gene rearrangement was occurring and we reached up to IgM, in the same way we are talking about light chain gene rearrangement. Okay, now this light chain gene rearrangement can be productive and can be non-productive also. Now productive and non-productive means successful and non-successful. If we are talking about the successful gene rearrangement, that means if the gene rearrangement was productive, the immature B cell is formed. Immature B cell is formed. Now we are calling that cell that was previously called as precursor B cell will now be termed as immature B cells. Now this immature B cell is having membrane IgM. I told you how membrane IgM was formed because of the previous events. Okay, because of the heavy chain rearrangement and the light chain rearrangement, you have got IgM finally. Now coming to the next step. So this was all of these things were occurring in step number 3. Previous to this, progenitor to precursor I have termed as step number 2, and from lymphoid stem cells to progenitor B cells I have termed as step number 1. Okay. Now let us move forward and talk about step number 4. Now once the immature B cells are formed, from the immature B cells we have to reach the mature or naive B cells. Okay, from immature we have to reach to mature or naive B cells expressing the MIGM, or expressing the so they are expressing the MIGM. It is having this membrane IgM and membrane IgD. But let us talk about how this step number 4 is occurring. So IgM is present. I told you IgM was present in the immature B cells. I wrote down over here only. So this was where I wrote that down. So IgM over here, on this immature B cell, IgM was present, plus there are two closely associated transmembrane molecules, and these are called as Ig alpha and Ig beta. That means along with IgM there are two closely associated transmembrane molecules that are called as Ig alpha and Ig beta, and remember that these two Ig alpha and Ig beta are linked together with the help of disulfide linkage. They have disulfide bonding in between them. Now this forms actually the B cell receptor. What forms the B cell receptor? Let us write down again. There is IgM, plus there are Ig alpha and Ig beta, and these are connected with disulfide bond. Together they form the B cell receptor. Okay, so what did we see in step number 4? We saw the process in which the B cell receptor is formed. Okay, and we also saw what it comprises of. Let us move forward and talk about that. After this process, what happens? So once the B cell receptor is presented on the B cell, now it is ready. Now it is ready in one way, it is ready to take on the antigens and everything. So what happens is, we are now calling it mature or naive B cells. Why am I calling them naive B cells? Because still they have not encountered any antigen. That is why I am calling them naive B cells. Okay, as soon as they will encounter any antigen, they would not be called as naive B cells. Now you would be knowing, if you have seen my one video that was on primary and secondary lymphoid organs, I have already said that the encountering of the antigen takes place specifically in secondary lymphoid organs. Okay, so now what will happen? They would leave the area of the bone marrow in order to get in contact with the antigens, and in order to gain specificity, they would leave the bone marrow as single and having single antigen specificity. Why am I calling it single antigen specificity? Because suppose this is my B cell. Now it has specificity against this antigen only, so it would be binding to this antigen only. It will not bind any other antigen. Suppose this is another antigen, it will not bind to this. It will only bind to this. That is why I am calling it as single antigen specificity, and this would be the stage in which the mature or naive B cells would be leaving the bone marrow. Now, after leaving the bone marrow, what is the way that they follow? They use blood or lymph, because these blood and lymph are the connective tissues, right? They would move here and there, and they would in some way or the other they would facilitate the relocation of these B cells to the secondary lymphoid organs. The purpose is to reach the secondary lymphoid organs. Now, in which secondary lymphoid organs basically we are talking about? So they can be spleen and they can also be lymph nodes. So we are talking about two secondary lymphoid organs primarily. One can be spleen, the other can be lymph nodes. Okay, so over here they reach. Let us talk about next step, that is step number 7. So I missed out on the steps. I believe you would have noted that down and understood that pretty well. Let us move on to the next point. Now, what happens in the secondary lymphoid organs? I have already said that secondary lymphoid organs are the places where the interaction between the B cells and the antigen take place. Now, once the B cell is over here, this is the mature B cell, but as of now it is naive. But once it reaches the secondary lymphoid organ, an antigen would also be present over here, and both of them will react with each other, and once they interact or react with each other, now the B cell will become activated. Okay, so B cell will now become activated B cell. Now when the B cell becomes activated, what will happen? Another process will start, that is called as clonal proliferation. Clonal proliferation. Clonal proliferation means making clones of itself, multiplying. Okay, why does the multiplication is required? Because in order to encounter the antigen, only one B cell would not be enough. So there has to be the copies of this B cell. Why there is requirement of the copy of this B cell only? Because you have this antigen and this antigen is recognized by this B cell only. That is why we have to make copies of this B cell, because it was having specific antigen property, right? So this will start multiplying. Okay, and after multiplication, some B cells would lead to the formation of plasma cells and some would form the memory cells. That means they would divide, not equally, but yeah, in some ratio they would divide into plasma cells and memory cells. Okay, now memory cells, what will they do? Memory cells, as the name suggests, it will form the memory. It will form the memory. What does the memory means? Memory means that when there would be a subsequent infection with that particular antigen, then the time required for the body to recoup and counter that antigen would be less because of those memory cells, and in those memory cells it would be saved. The information would be saved that how we tackled this kind of information. These B cells would be these B cells would be representing the event that had taken place previously, and now these B cells would become active after subsequent infection from that antigen, and they will start proliferating, and there would be less time required for this immune response as compared to the first immune response. I believe this makes sense. Now let's move to the plasma cells. Now these are, I believe, more well understood. Plasma cells. Now plasma cells are the cells that have the capacity to form antibodies. These plasma cells are also called as antibody secreting cells. Plus it has been seen that they can secrete antibodies around 2000 molecules per second. 2000 molecules per second are secreted from these, I mean to say antibodies are secreted from these plasma cells. Next important feature is that these antibodies are targeted against a single epitope. They are of a single Ig class and they are having single light chain. Single epitope means, suppose there is this one antigen and it is having multiple epitopes. So this was one epitope, this was one epitope, this was one epitope. There are multiple epitopes on this antigen, but the antibodies that would be targeted would be against one epitope only. So suppose this epitope is targeted. Okay, so all the antibodies that would be produced by one plasma cell would be targeting one B cell would be targeting one epitope. They would be of a single Ig class. That means it can be of IgA, IgG, any class, and it would be having a single light chain. That means there would be no mixture of the light chains that we had talked a little while ago. So these are the important features of B cell development and differentiation. Now let us talk about the diagrammatic representation. Now I have taken this diagram from a Pathfinder book, okay. So let me explain this diagrammatic representation. Okay, so this was the stem cell where we started, lymphoid stem cell. From the lymphoid stem cell, remember we had reached on to the pro B cell. In case of pro B cell, there was heavy chain gene rearrangement. Remember, Ig alpha and Ig beta are present over here and they are linked up with disulfide linkage. So there would be some disulfide linkage in between both of these. After this, there was heavy chain rearrangement, right? Heavy chain rearrangement. Because of the heavy chain rearrangement, you have over here mu chain. Because of the mu chain, in future it will be forming the IgM, that is written over here, just after the light chain rearrangement takes place. Now over here surrogate L chain is mentioned. I believe you remember surrogate L chain, we had also talked about this. This is present over here and you can see that it is represented by dotted lines. But as soon as the light chain rearrangement takes place, this is now shown by one complete line. Light chains are shown by one complete line. After this, this was the immature B cell. After this, mature B cell is present, and in the mature B cells you have membrane IgM, plus this is the membrane IgM, this is the membrane IgD also. Remember, this is IgM and IgD, both are present in mature B cells, and there is also Ig alpha and Ig beta. Okay, so this was forming the actually B cell receptor, I told you. So this everything was happening in bone marrow. Everything was happening in bone marrow. This was the antigen independent phase. That means this phase was not dependent upon antigen. Up till now you did not see any antigen coming over here, right? There was no antigen. But now the mature B cell would be transported to the lymphoid organ, secondary lymphoid organ or peripheral lymphoid organ. Now this was the mature B cells, IgM and IgD were present. When they encounter the antigen, and because of the TH cell, what happens is these mature B cells become activated B cells. I told you. After that there would be clonal proliferation, or you can also call that as clonal expansion, and then they would be divided into plasma cells and memory cells, and they would be secreting multiple antibodies. This is what I had shown you a little while ago. One more important thing that I missed out was that mature B cells are the cells that are having membrane IgM and membrane IgD. Remember that both of these IgM and IgD are present, and they are in turn forming the B cell receptors. Okay, IgM is also present, IgD is also present, and Ig alpha and Ig beta are also present. So I believe this made things clear. I would lastly show you the diagrammatic representation again. I believe this would be clear to you. So if you have any questions, you can directly post on the comment section. If you like the video, just hit the like button, subscribe to my channel, press the bell icon, and also share with your friends so that you all can learn more. Thank you and have a good day.